If young Metro don't trust you, I'm gonna shoot you. Beautiful morning. Yeah, the sun.
Before we get into the process of humoral immunity, I'd like to talk to you about B cells. B cells largely comprise of what humoral immunity is, and the maturation of B cells and their turning into antibody-secreting plasma cells is what humoral immunity is mostly about. As the response of as the response of B cells decline, they leave behind a pool of memory cells which memorize and remember which antigens were present before, and they'll know to attack those antigens again. We begin with the process of humoral immunity. There are 5 steps in humoral immunity, and they are antigen recognition, clonal expansion, differentiation, elimination, and last, memory.
So that many get plenty, many get plenty, many get plenty. It's goes in that back and all they know is gimme, all they know is gimme, all they know is gimme.
We begin humoral immunity with antigen recognition. Antibody receptors are capable of responding to many different chemical structures such as proteins, lipids, polysaccharides, small chemicals, and etc. This is a B cell antibody receptor, and this is a B cell. An antigen circulating within the body only initiates a humoral response if it randomly encounters a complementary antigen receptor with high affinity, meaning this red particle right here, which is known as an antigen, if it were to meet with this receptor and that receptor were to have a high affinity for this antigen, only then would that antigen initiate a humoral response from that B cell. B cells recognize antigen via membrane-bound antibodies. These antibody receptors are capable of recognizing many different chemical structures as mentioned before, proteins, lipids, polysaccharides, and small chemicals, etc. B cells can recognize antigens that are circulating freely in the blood or that are attached to the surface of microbes. This is a key difference between B lymphocytes and T lymphocytes, as the receptors of T cells are only capable of recognizing small peptides that are presented to them by antigen-presenting cells. Antigen-presenting cells are a special kind of cells that, in a sense, grasp antigens and carry them to a B cell where they can initiate a humoral response. The second step of humoral immunity is clonal expansion. The process by which an antigen activates a single type of B cell is known as clonal expansion. The body generates billions of different lymphocytes that can potentially recognize millions of antigens throughout the body. Although there are a myriad of different lymphocytes, since there are billions of cells in the body, there's a good chance that there will be copies of lymphocytes that have the ability to recognize the same antigens. The thing that makes these specific lymphocytes similar is that they both have the same antigen receptors, and this enables them to seek out the same antigens throughout the body's circulatory system. These receptors are passed down genetically, enabling for immunity among generations of lymphocytes. This sect of lymphocytes with the same receptor is known as a clone meaning a unit. Thus, when an antigen activates 1, 2, or even 50 of these clones, it is said that the entire clonal unit is activated. Once these are known to be activated, the cells in the unit begin to divide, and this is known as clonal expansion because the cells start to expand more. Over here, you see different types of cells, being one clone, another clone, and another clone. However, this clone has a immune response for this antigen, and what happens is, once this antigen is encountered, this clone starts rapidly duplicating these B cells in order to compete with these rapidly duplicating antigens. And this is supposed to keep pace with that so that this can eliminate that. Now we come to the third step of humoral immunity, and that is differentiation. Once a clone of B cells have been activated, 2 key things are happening. First, the cells divide. By increasing in number, the clone ensures that it will have enough cells to mount an effective immune response and that its numbers will keep pace with the replicating microbe. Second, the cells differentiate into plasma cells, which begin producing and secreting antibodies that are specific for the original antigen. If the antigen is a protein, it will be phagocytized by antigen-presenting cells and displayed to T cells. This activates helper T cells and causes them to release mediators to improve the B cells. In fact, helper T cells are essential in mediating an effective B cell.
Shit don't do.
Fourth step of humoral immunity is antigen elimination. Antibodies are the effector of humoral immunity. Antibodies are capable of neutralizing antigens, marking them for destruction by macrophages, and activating complement and instigating vigorous responses from the innate immune system. In addition, antibodies confer passive immunity to neonates while their immune systems mature. There are several different types of antibody isotypes, such as IgA, IgG, IgE, and others, etc. And each has its own mechanism for neutralizing and eliminating antigens. As the humoral immune response winds down, some plasma cells will undergo apoptosis, which is cell suicide. The others migrate to the bone marrow, where they will continue to secrete antibodies for years at a time. Here you see the first step, neutralization. The antigen can no longer cause damage because it's now physically bound by one of the receptors. Second, this second part is called opsonization and phagocytosis. This antigen is marked for phagocytosis and destruction, so it will now be consumed by the cell via cellular phagocytosis. And the activation of a complement, which basically means it picks which of these actions it want to take. This is a phagocytosis, where it will consume the cell. This is where it swells up with water to help isolate the cell. And this is the lysis of the microbe, which is basically self-destruction of the microbe from the inside.
I got broads in Atlanta, just to lean the family, credit cards in the scammers.
The fifth and final step of humoral immunity is memory. The initial activation of naive lymphocytes also stimulate the proliferation of long-lived memory cells, which may survive for years after an infection. This pool of memory cells is much larger than the pool of naive lymphocytes specific for any antigen that is present before the encounter with that antigen. In addition, these memory cells respond faster and more effectively against the antigen than naive lymphocytes. The generation of these long-lived memory cells is one of the primary goals of vaccination, which helps put in cells that immediately recognize what the pathogen or antigen is, isolate by attaching to it using the receptors right here, and committing one of the processes of antigen elimination, such as swelling, lysis of the microbe, and or phagocytosis. Generally, cells that would do this are white blood cells.
Open up the champagne, pop. It's my house, come on, turn it up. Hear a knock on the door and the night begins. Cause we've done this before, so you come on in. Make yourself at my